speaker tonight is Katerina Graham. And Katerina, to say the least, has a very interesting background. She's worked as a naturalist, environmental educator, instructor for gifted and talented ISD programs, environmental educational programs manager, and a biology and Spanish teacher. Most recently, she worked for the USDA's Agriculture Research Service in Corvallis, Oregon, as a research assistant project manager for biocontrol studies. Then in 2022, she joined Texas A&M's AgriLife Extension Service, where she served initially as a horticulture agent for Dallas County, and most recently, Katerina's transitioned to the Dallas Research and Extension Center, where she currently works for the Controlled Environment Entomology Program. So with that, we'll transition over to Katerina.
Good evening, everybody. My name is Katerina Graham, and I am very pleased to be here tonight with you. I just want to give you a full disclosure that although I have worked with parasitoids in the past, I am not an expert, but I was very interested in creating this presentation for you because I really have gotten to really appreciate and be amazed by these little creatures. And there are so many of them, and there is so much to know that I think that I have 20 years ahead of me to learn more about them. So I just wanted to share a little bit of what I have learned with you tonight. And it will, I believe, last about an hour. So if you do have questions, so that you don't forget, please put them in the chat. And if I don't get to them tonight, I believe that Tim will email them to me, and then I will be happy to answer them. And so we will get started. Here we go. So parasitoids, the first thing that I will point out is that you notice that word. It doesn't match the typical word that we hear, parasites. And that there is a reason for that. It's not the same thing. A parasite has to keep its host alive in order to keep itself alive. Whereas a parasitoid will invade the body of the host and sometimes will keep it alive for a little bit, but at the end of the day, it will kill that host. So a parasitoid is kind of a combination of a parasite and a predator because it does kill it. So what we typically see with parasitoids, here you see the typical life cycle of one, is that the adult female will lay her egg into a host, and it can be on top, on the outside, it can be on the inside, or it can be near the body of the host. The eggs will hatch, and the developing parasitoid larvae will consume the host, and it will sometimes turn into a pupa inside. Sometimes they will do it outside, and eventually the adult will emerge and look for another parasitoid of its species, mate, and the whole cycle starts over. What's incredible is that really of the estimated 4 to 8 million species of insects, a whole 1.5 to 2 million are parasitoids. So there's a huge number out there. It's just that we don't see them because they're so tiny. Some of them can be smaller than 1/16 of an inch. And so the only time really that you get to see them is if you put a sticky trap outside, and you will catch a bunch and you can put them under the microscope and see them. The word parasitoid as such was coined by a gentleman with the name by Reuter at the beginning of the century. And what's also important to know is that although today I am going to be emphasizing the hymenopteran parasitoids, this lifestyle has evolved independently many times. And so we can find parasitoidism in Coleoptera, which are the beetles, Diptera, which are the flies, Lepidoptera, moths and butterflies, Trichoptera, which are the caddisflies, Neuroptera, the net-winged insects, and then also Strepsiptera. See if I can get that out. So, but again, I'm not going to talk about those. I'm just going to talk mainly about Hymenoptera tonight. So once again, when it comes to the reproductive cycle of parasitoids, it's very common to find those that are relying on the larva or the nymph of another insect. I think most of you are familiar with some of this vocabulary, but nevertheless, I'm going to pause and just remind some of you that there is a difference between a larva and a nymph. A larva usually refers to the young that emerges from an egg, and we are talking about those insects that go through the complete metamorphosis, so egg, larva, pupa, and adult. Those are the ones that we call larvae. Nymphs are also young that come out of the egg, but they are part of the group that goes through incomplete metamorphosis. And so those young already look like the adult. They're smaller, they don't have wings, and they slowly develop into the adult stage. So again, going back to our parasitoids, it's very common to find parasitoids that will use both the nymph and the larva. It's also fairly common to find parasitoids that will parasitize the eggs and pupa, and not so common, not so common to find those that parasitize adults, but we do find examples of them, and we'll talk about one of them towards the end of the presentation. This is a cladogram. You may be familiar with what a cladogram is. Basically, don't get lost in the details. I'm going to point out some general things here, but a cladogram is basically this graph that depicts the evolutionary relationships between organisms. And so when we think about Hymenoptera, we're probably usually thinking about bees and bumblebees, right? And so just wanted to show you the immensity and the biodiversity that exists in Hymenoptera. And those bees and bumblebees are mostly right here where I have put that little oval. So they're in the Apoidea group. And right next to it is the Formicidae, which are the ants. I'm going to highlight two more, Pompilidae and Vespidae. Vespidae being the true wasps, Pompilidae being the spider wasps. And the reason that they have that orange line is because, as you can see over here on the, gosh, I'm forgetting here the name of this, but it's helping with the key. It's helping us know that these particular groups are the nest provisioners. Something else that I want to point out in this cladogram is, as you can see, some of the groups are extremely wide in terms of their geometric form. And what that is representing is the biodiversity. So for example, if we look at Chalcidoidea, you can see that it's a very biodiverse group versus something like the Braconidae or maybe the Proctotropomorpha. So those are much, much smaller. And being that they are in the red color, that means that they are parasitizing either the larva, the pupa, or the adults. The ones in pink are the ones that are parasitizing the eggs. So you can see that there are not that many of them. And then finally, some of you may have come across when you're out and about with the gall wasps, and most of those belong to Cynipodiae. So just again, I just wanted to put this up so that you can see how diverse the parasitoid world is. Okay, so moving into the strategies, parasitoids can either lay their eggs on the outside, so that would be an ectoparasitoid, or they can lay their eggs on the inside, and that would make it an endoparasitoid. Both have their pros and cons. An ectoparasitoid does not have to worry about, how it's going to breathe or whether or not the immune system is going to attack it, but it does have to worry about maybe being eaten by a predator that sees it on the body, or maybe in this case, if this spider molts, it may fall off the body of its host. And so it does have to contend with certain challenges. The endoparasitoid, on the other hand, is protected from the environment because the egg is inside of its host, but it has its own set of issues to deal with. That being that now it's harder to get oxygen into its own body. It will be attacked or try to be attacked by the immune system of the host. And so each one of these has developed a way to, has adapted a way to overcome that challenge. Now, because this is nature, there are also some hybrids of this. And so here is an example of one that starts out by being on the outside. This is the emerald jewel wasp. It is known to attack the American cockroach. So the egg starts on the outside, but as it emerges from the egg, that larva will go inside of the body. And we'll talk a little bit more about the emerald jewel wasp in just a minute. And then we have the opposite. We have some that will be laid into the host, and then at some point in the development of the parasitoid wasp, one of the stages will come out and be on the outside of that body, as you can see in this tomato hornworm. Okay, so I want to kind of jump back into these challenges, especially that the endoparasitoids have. As I said, the ectoparasitoid, basically, it just uses its standard tracheal system to get air. Here is a pretty cool picture of a larva that you can actually see through its epidermis, its exoskeleton. And so just a reminder of how an insect gets its oxygen. It has these small openings that are called spiracles. Those are open out into the environment, and through them, oxygen can pass and go into the trachea, and then through diffusion, it goes into the tissues and the blood of the insect. So that is how a regular insect breathes. Now, if the egg of a parasitoid has been injected or laid inside of one of these hosts, now it has a problem because it cannot reach that oxygen that is just free floating in the environment. So there are two options that it has, or that have been developed. One is to use dissolved oxygen that is floating around in the hemolymph, which is the word for insect's blood, or it can hack into the tracheal system of the insect. So once again, here is the emerald jewel wasp. And basically what happens, maybe I'll give you a little bit of a description of what goes on with this particular relationship. The female of the jewel wasp will sting the cockroach twice. Once it will put venom into the tracheal, I'm sorry, not the tracheal, the thoracic ganglia. Ganglia basically is this just a big mass of nerves that come together that help to control functions. Uh-oh. Are you still there? Oh, no. Hello?
We're still here, but yeah, we lost your presentation.
Okay, let me see what happened.
We got your video. We still see you.
That's weird. All right, let me try again. Well, go figure, it's gonna crash on me. IT, you have to love it, right? Oh my goodness. Let me see what happened.
Do you have the Zoom? Do you have the Zoom menu
Yeah.
to share again?
Oh my gosh. My computer decided to update right now. Are you kidding me?
That was great timing by it.
Well, I'll tell you what, while this is going on, I'm going to reverse the presentation and I'm going to talk to you about biocontrol, which is what I have been involved in. Let's see, here it comes. Oh, maybe it's going to come up sooner than I expected it to. All right, let's see. Oh, Lordy. I'm just going to laugh so I don't cry. Okay, so where were we? Here you go. And next slide. Can you see this?
Not yet.
Not,
There it is. Oh, well, see Carol.
not yet. Okay. Let me see. Maybe I have to do this again. So Tim, you may have to share with me again the host privilege because I don't.
Oh, okay. Let's see. Yeah, yeah, it still shows you as a co-host. I don't know what happens if I go and make you the host. And I think we'll stay with the co-host. Pardon? All right. Good thought.
Okay, I see.
There, I re-added you.
Okay. All right, so can you see?
Yep, we can see it. Thank you, Melanie.
Can you see it? And let me try to get it up completely. Can you see that? Is it showing the full
Looks
screen?
good. Full screen. Great.
Yes. Okay. Okay, so let's continue. Let's bring the emerald jewel parasitoid. Okay, so I was at the point that it had injected venom into the ganglia, and that is so that the cockroach will stop kicking and so that the wasp won't get hurt. Once that happens, it will approach the brain areas and inject a second round of venom. And what that does is basically just make the roach very compliant. Once that happens, the jewel wasp will move away from the roach, look for a burrow. Once it has found one, it comes back to the roach, it chews off its antenna, feeds off the hemolymph that's coming off the antenna, and then the little stubs that remain on the cockroach are basically like a leash that the jewel wasp can grab, and almost like walking a dog, it will take the roach into the burrow. And then it will lay the egg in between the middle legs of the roach. Once it has done that, it will abandon the burrow and just kind of secure it so nothing will be a predator on its young. Once that egg hatches in a few days, maybe 3 to 6 days, the larva immediately, it has mandibles, and so immediately it'll start chewing into the cockroach. And the first system that it chews into is the tracheal system. As soon as it does that, the tracheal system starts to release small bubbles of air, and the larva will make sure that it lines up its own spiracles with that trachea and where those air bubbles are being released. And so that is how that particular parasitoid overcomes the challenge of breathing inside of the roach. So it's a crazy, fascinating story, but let's move on because there are more challenges. The second challenge is to resist the host immune system. So here again, there are 2 ways that parasitoids have, or more than 2 ways actually, that the passive and the active. Before I move into this, let me give you a little bit of background about immune system. Just like humans, insects have these cells, they're called hemocytes, that will seek and attack a foreign body inside of their own body. And so once they do that, they will actually, these hemocytes will sort of gather on the outside of that foreign body, basically encapsulating it. Once it has encapsulated it, it's going to start to create this scab. This is a process that's called melanization. And during that melanization time, these very powerful toxins get released, and that is what will kill that foreign object. So if a parasitoid does not want its egg to be recognized as a foreign object, it needs to protect it. And it actually does that. It covers it with some proteins that will eventually make it, hide inside of the body. So what I'm going to show you right now, this first set of pictures, you can see the larva of a fly, and inside of it, you can see this encapsulated wasp egg. A closer look shows you the fly hemocytes that are starting to wrap around that wasp egg. But you will be wondering, well, Katerina, didn't you just say that that's not going to happen? That is correct. It shouldn't happen. And the reason this is happening is because the scientists that took these pictures actually removed the proteins from this egg and then added the egg artificially into the larva, just so that they could take these pictures. But normally this would not occur because that layer of protein that's coating the egg basically makes the egg disappear. Other parasitoids that don't have this passive system will lay their egg either onto the brain or the ganglia or fat bodies. And the reason is that hemocoel doesn't really cover that area extensively. And so the eggs will not be found. Or the egg. Sometimes it's just one, and we'll get to that. Here is the active adaptations. The first one is that when the wasp, the female, lays her egg, with that egg, she's injecting venom. And that venom basically disables the hemocytes. So they are no longer able to adhere to the foreign body, which in this case would be the egg. So that's one strategy. The second strategy is there's a group that the Icmonidae, they have this symbiosis with viruses, specifically what is called the poly-DNA virus. And so this virus is delivered with the wasp egg, and what it does is it does not attack the caterpillar or the larva or the nymph. What it does go into the hemocytes and triggers what is known as apoptosis, which is this fancy word for cell death. Therefore, those hemocytes cannot encapsulate the egg, and the egg can continue through its normal development. So 2 ways that the parasitoids' eggs can resist the immune system of a host. Okay, let's talk about killing the host. There are 2 categories. One of them, which is known as, they are known as the idiobionts. They actually will lay their eggs, and that host will be immediately consumed by the larva as soon as it hatches out of the egg. Most of the times, it will attack the parasitoid will attack a host that is immobile. So it can be either an egg or a pupa. And as you can see here on the slide, they tend to be generalists. This little guy here on the slide is known as Trisulcus japonicus, or TJ. They're also known as the samurai wasp. And you can see it she's laying her eggs on the brown marmorated stink bug eggs. So as soon as those little eggs hatch, the larva will just go ahead and kill those brown marmorated eggs. They will not last long. Now, a different category, which are known as the coinobionts, that is entirely different. Instead of killing the host immediately, the host will be temporarily paralyzed, and then the host continues to feed, to develop, to get larger, to get stronger. And it is only when the host reaches a certain size that the parasitoid larva will continue its development. So again, once the parasitoid egg is laid, the larva will actually hatch, but then its development stops and it waits until its host gets larger. And this is a benefit to the female parasitoid because she can lay her eggs into very small creatures when she finds them. But that small larva can take advantage of, just it benefits from waiting so that it has enough resources to complete its development. And so once again, you see a tomato hornworm here with the Catesia congregata, which is the one that usually lays many eggs, and you can see the pupa coming out of its body here. And again, Trisulcus japonicus is above it. Okay, so social preference. This is from the standpoint of the larva, not the adults. So many parasitoids will develop only one larva per host. So they are considered solitary. In some cases, a second larva will be laid into the same host, but if the solitary parasitoid was the first one to lay her egg, that larva will already have emerged from the egg. And again, they do have mandibles. And so if there's a second one that hatches out, it will be attacked and eaten. It wants to be by itself. It wants the host just all to itself. The other kind, the gregarious parasitoids, again, from the standpoint of the larva, we can have situations like the one we have seen now several times with the tomato hornworm, where multiple eggs are laid on or inside of the host, right? And so you tend to have dozens and dozens that come out. And it's because dozens and dozens of eggs were laid into the host to begin with. Or we can have a situation that is known as polyembryony. And that is a little different. In this case, the parasitoid, the female, lays only one egg. And once that egg begins its development, it starts to turn into a number of embryos. And sometimes, as you can see in this picture down here, it can really be extremely fertile, if you wish, or productive. You can have several hundreds to several thousands of larvae that come from that one egg. And so that would be considered a gregarious parasitoid. And in this case, the Copidosoma genera has this example of polyembryony, and it has also an example of a different strategy that we'll touch on in a few slides from now. Okay, so now to competition for resources. One of the things that I want you to keep in mind is that not all females have this ability to discriminate whether or not eggs have already, or not eggs, but whether a host has already been used or not. If the female host can discriminate, then she's probably making use of chemical marks that were deposited by the first female that was there. But again, not all parasitoids have this ability to discriminate. So if they do not have that ability to discriminate, several situations can arise. The first one is what is known as superparasitism. So this is a female, again, that cannot discriminate. So she will add, the second female will add her own eggs to a host that is already parasitized with an individual of the same species. So that is what's considered a superparasitism. If in a very similar fashion, if a second female parasitizes a host, but if the first parasitoid was from a different species, that is known as multiparasitism. And then there's a third situation that can occur, and that is called self-superparasitism. This is where the first female already parasitized a host, and then she goes about parasitizing things, and then she comes back to the same host or the same egg or larva or pupa. She cannot tell that she has already laid an egg into that host. So that is known as self-superparasitism. Whether or not multiple will develop will all depend on whether it's gregarious or it's solitary, or it also depends on how far along the first parasitoid larva has developed. But usually they will be, the second one will be attacked. Now, the level up from competition for resources is basically feeding on other parasitoids, period. And so this is very widespread. It is known as hyperparasitoidism. Many of the hymenopteran parasitoids tend to do this. And here on this graph, you can see the food chain, right? So we can start down here with this brassica of some sort. Let's call it a kale. And we can see, maybe we'll just follow one line, that Pieris brassica is feeding on the kale. Catesia glomerata comes and deposits her eggs on Pieris brassica. And then Lysibia nana detects the fact that Catesia glomerata is inside of that larva, seeks it out, and lays her eggs into the larva of Catesia glomerata. So that is what is known as hyperparasitoidism. In some cases, there is what is known as obligate hyperparasitoidism. So it has to be specific from a parasite, from one species of parasitoid to another species of parasitoid. And unless that second parasitoid can find the first one, there won't be any reproduction. Or there are some species that have more of a facultative type of hyperparasitoidism, which means that if they come across a primary parasitoid, they will lay an egg into it, no problem. If they don't come across a primary parasitoid that's already inside of the host, they will just go ahead and lay their egg into the host. So they're not picky, and they don't need a specific species for their young to develop. Okay, so that is the difference between obligate and facultative. In some cases, there are some examples of parasitoids wherein the same species needs to lay an egg on itself. So this is true for the aphelinids. In some cases, the females develop in one way and the males develop on a female of its same species. So I know I'm probably blowing your mind at this point, but I'm going to keep going because there are many other examples of these crazy life history strategies. I did want to include this slide because as I was preparing for this presentation, I came across this report that just came out last week on the Rice University campus in Houston. The scientists or the professors there on the Rice University campus found and have reported this brand new hyperparasitoid of the gall wasp, Neuroterus bucei. Neuroterus is a gall wasp of the southern live oak, And so this new hyperparasitoid, Chrysostomia subcelli, is the first of its kind worldwide that has been reported on this type of gall wasp. And so I thought that was pretty cool that in our own backyard, so to speak, we're still finding new species. And scientists do believe that we will continue to find new species of parasitoids, again, because they have, in some cases, not only are they tiny, but I described what would be considered a secondary parasitoidism right on this slide. But there are reports of tertiary hyperparasitoidism, quaternary, and even a fifth level. So that is why it is believed that we really have not discovered a large fraction of all of the hyperparasitoids that are alive today. Before I move on, I have a couple of video clips that I'm going to share this as a PDF. And if you want to see further some crazy life histories of parasitoids in general, you are welcome to watch those at your own time. I figured we would go over time if I showed you them. So I hope that you take advantage of that and watch them. Okay, now there's a different example. We talked about hyperparasitoidism. In this case, we have a situation where the parasitoid needs multiple hosts, okay? So this particular one is Encarsia porteri, and the female will lay her eggs on this pupa that belongs to a white fly. That's what the little white fly pupa look like. I work with these at the Dallas Center Research Center. And just again, as a little bit of background information, or to remind you if you already know this, hymenopterans have this ability to decide what gender their young are going to be. It's called haplodiploidy. And so if the female E. porteri wants to have female young, she will fertilize it with the sperm that she's carrying in her body. And she will seek out these white fly pupa. But once she decides that she needs male offspring, she is going to go off and look for a different host. In this case, these are Lepidopteran eggs, and she will not fertilize those eggs. So they are unfertilized eggs. This is not widespread throughout the hymenopteran world. And in fact, I believe that there's only one other order where this type of strategy is found, and that is in the strepsipterans. They also will do females on one species and males on a completely different species. Okay, so remember that I told you we were going to come back to this genera, and it's because not only do they have polyembryony, but in some cases, a different species has something that is known as polyphenism. So again, from one egg, many embryos will develop, but not only will they, in this case, some of them will be different. So there's almost like a caste system that develops. Some of those larvae that you see in that on the picture on the right-hand side, inside of the larva of this, I believe it's some sort of lepidopteran, will be soldiers, and then some of them will be reproductive adults. So the larvae that are soldiers are just kind of wandering around, making sure that every larva that is in that body belongs only to its own species. If it finds a different species, it will attack it and eat it. In some cases, it's not known how they know this, but if there are too many male larvae inside of this host, the female soldiers will also attack those male larvae, because the more resources that the females have, the more offspring that particular line will end up with in the long term. So many crazy strategies. There's one more that I want to point out, and this is something that's called phorecy. A phoretic behavior is considered when one organism kind of hitches a ride on another, okay? And so in this case, here you can see a cabbage moth. You can see her face, and right there you can see a little parasitoid. It's just getting a ride to wherever this moth is going, right? The reason it's considered phoretic and it's not considered parasitic at this point is because the parasitoid is not harming the moth in any way. Just to give you kind of some perspective, if a flea that is on your dog is also hitching a ride, but at the same time is feeding off your dog, that is not considered phoretic anymore. Okay, so it's phorecy only when it's just being transported. Now, of course, the parasitoid is hitching a ride because it knows that whenever the moth gets to its destination, it's going to be laying eggs. And so at that point, it will detach from the moth, and through several cues that it can detect, it will find the eggs and go ahead and deposit its own eggs. Here is another example. Here we have some parasitoids that are actually riding on the ovipositor of a bush cricket. And so not only are they hitching a ride on the body, but they're hitching a ride exactly where those eggs are going to come out. And it's because the chorion of the egg of the bush cricket tends to harden very quickly. And so they want to make sure that they're there to lay their eggs before that chorion hardens. Okay, back to virus symbiosis. We talked a little bit about the symbiosis that allows the poly-DNA virus to, in effect, stop the hemocytes from encapsulating any type of foreign body. This is a different example of a virus that helps the parasitoid. This Dinocampus coccinelliae has this virus inside of her that she will inject into the ladybug when she injects her egg into the ladybug. This picture you see right here, she actually does not lay her egg into the brain of the ladybug. Right now, she's just paralyzing the ladybug, similarly to what the emerald jewel parasitoid was doing. So they sort of paralyze the ladybug, then she will crawl around, get close to her abdomen, and insert her egg through the abdomen of the ladybug. The egg of the parasitoid will hatch, the larva will begin to develop, the ladybug will continue her own life and eating and doing whatever ladybugs do, until that larva of the parasitoid is ready to come out. At the point that the larva is ready to come out of the ladybug, that larva will release some chemicals that again affect the brain of the ladybug. And the way it affects it is that as the larva of the parasitoid of Dinocampus comes out and begins to turn into a pupa, you can see that pupa starts to develop actually on the legs of the ladybug. And the reason that happens is because pupa are, they're exposed to the environment. So predators could easily come and feed on them. So not only is it being sort of protected by being covered by the ladybug, but remember, those chemicals that were released sort of turns that ladybug into a zombie. And that ladybug no longer feeds, it just takes care of that pupa as if it was its own child. It's almost like a bodyguard. And so that allows the any predators that will come near it, the ladybug will attack it, will defend the pupa. And once that pupa finishes its development and the parasitoid leaves, in many cases, that ladybug will survive. And of course, those substances will metabolize and the ladybug goes back to being its regular ladybug. So it's incredible, really, how much of the evolutionary tree can just, it's there are so many examples of these very specific interactions. And again, that's why I was saying I could take forever. It's going to take me another 20 years to learn all of them. But host location, I have 10 more minutes. These, as I mentioned to you, parasitoids are extremely tiny. And they are in this huge environment of ours. They are out in the fields, they're in the forests, and they are also looking for, in many cases, very small insects. And so you can imagine it is not a very easy endeavor to try to do that. However, hymenopterans and parasitoids in general have evolved and adapted over eons, and so there are quite a few ways that they can find their host. The most obvious stimuli that will point to a habitat or a microhabitat will be something like, if the parasitoid is after a caterpillar, it will kind of direct itself towards something that's green, right? But let's say that the parasitoid is the parasitoid of a fruit fly. It's green is not going to be it. Instead, it might use the smells of things that are fermenting, because, as we all know, fruit flies love anything that is decaying, because that's where they lay their eggs. And so those obvious stimuli are the first kind of level that a parasitoid will use. The second one that they tend to use quite a bit are known as HIPVs, which stands for herbivore-induced plant volatiles. When a plant gets attacked by a predator, a predator, which would be, for example, a caterpillar, they will release these plant volatiles. And in some cases, they can be very specific to parasitoids. So again, it's they've co-evolved. And so it's the parasitoid is just waiting for those HIPVs that are specific to it to come up in the environment, and it will target that particular plant. They can also direct their attention to the host's wastes. It can be frass or poop. It can be their saliva. It can be mandibular secretions, silk, or even scales. So that cabbage moth that we saw a little while ago, when she lays her eggs, she probably, a few scales fall off her wings, and that is what that parasitoid is cueing into. In some cases, there are bacteria or fungi. These are known as symbionts that are in a relationship with these host organisms, and they themselves, the bacteria and the fungi, will release chemicals, and the parasitoids have adapted to pick up on the symbionts' chemicals that are being released. In some cases, it's pheromones from the insects. So for example, bark beetles, they tend to aggregate before they attack a tree. Why? Because they want to overpower the tree's immune system, and they do that by descending on the tree in large numbers. In order to attract their fellow bark beetles, they have to release this aggregation pheromone. Well, guess who else is in tune with that aggregation pheromone? The parasitoid of the bark beetles. So that is good and bad for the bark beetles. And so it's just, it's a free-for-all out there. Sex pheromones, they can also be in tune with that. It is known that there are some parasitoids that will actually not be in tune with the sex pheromone, but they're in tune with the sound of the stridulation that crickets make to attract their mates. So they're actually using a sense of hearing or vibration. So vision, vibration, thermal, all of these are used by parasitoids, especially if their host tends to be a concealed type of host. Okay, so just to finish off here, I wanted to bring in the biological control part of it. Again, this is what I'm involved in right now at the Dallas Research Center. We are studying whiteflies and trying to determine if there are interactions between different natural enemies. If you're not familiar with biological control, basically what it means is that we are taking advantage of the fact that pretty much every organism has a natural enemy, or maybe two, or maybe three. This can be a predator, it can be a parasitoid, it can be a nematode, it can be bacteria, fungi, mites, it can be any number of natural enemies. And so we are trying to determine what kind of interactions occur between natural enemies, so that, for example, if they want to, something needs to be released in a greenhouse, but it's not quite controlling the pest, which in this case would be the whitefly, that maybe we know that we could add a second natural enemy, and that natural enemy will also go after the pest and not necessarily be a predator on the primary predator. So those are the kinds of studies that we will be conducting. It's a new program. But back to biological control, it's generally regarded as three distinct types. You can see here the classical, the augmentative, and the conservation. The augmentative is basically for immediate impact, is what the greenhouses are using, or maybe an individual small field. It's usually meant for a single generation of a pest, especially if a lot of them are released at the same time. So if many, let's say hundreds of parasitoids are released at the same time and sit inside of a greenhouse, that is considered inoculative. In some cases, I'm sorry, let me back up. That's considered inundative, right? It's kind of like a flood of parasitoids. Inoculative is more of a prevention type of a release. So let's say that I'm running a greenhouse and I know that it's early February, March, and I know that aphids tend to come out the third week in March. Well, I want to get my population of aphilinids ready, and so I will inoculate my greenhouse with them and hope to time the release so that right when the aphid population is peaking, that those parasitoids will be in place to control their numbers. Conservation biocontrol is probably what you folks would be interested in most, because it has a lot to do with native plants, native bushes, native grasses, and especially around in this area, of course, because all of this used to be prairie. But you can see, and I'm not going to go into each one of these, but the point is that we can not only attract, but we want to keep the parasitoids in the area where we are either growing edible crops, or maybe we have ornamentals, or maybe, and even weed control. Sometimes weeds are being controlled with the use of biocontrol agents. But we don't want to have the situation where we're having to release them and buy them from companies over and over and over. We want to make sure that we're providing plants that will offer nectar and pollen, because the adults, the females of parasitoids, they many times will feed on these plants. And so this is one way to keep those populations of natural enemies in place, in situ. And then finally, the classical biocontrol, this has to do with trying to control those invasive exotic species, whether we brought them in by accident or we brought them in on purpose. We have lots of invasive exotic species in not only in our country, but other countries have exotic species that don't belong there. And so because they don't belong there, they don't have their natural enemies. And so what scientists will do is try to pinpoint the origin, the geographical origin of that invasive species. They will send scientists to try to track down natural enemies. Once they have figured out, or they think they figured out which natural enemies will work, they will bring them back and conduct studies for many, many years. The EPA now requires 15 years of studies before they will approve the release of a natural enemy that was not that did not originate in North America or in the United States. That is something else that I was had the privilege of being involved in when I worked in Corvallis, because we were releasing a new species called Ganaspis brasiliensis that came from China. And it was basically so that it would control or reduce the population of the spotted wing Drosophila. So that's the Drosophila suzuki. We don't have that problem here in Texas, but on the Pacific Northwest, it's a huge issue, especially for soft fruits like blackberries, raspberries, strawberries, etc. That is classical biocontrol. One more slide here, and this is just to kind of show you that the differences between them mostly have to do with the spatial and temporal scale. So once again, inundation is a local and short term, it's fast, compared to importation or classical, where we're trying to bring in something from the outside, and we really have to do we have to release these across large regions, and we're hoping that it will stay long term so that we can actually control the pest that we are dealing with. So there you have it. There is one more link right here, and that will be another interesting life history of a parasitoid. But if you have any questions, there is my email. And thank you so much. I hope I did not go over time too badly. And I will maybe take questions. You can let me know, Tim.
Yes, thank you so much, Katerina. Fascinating. Very good. So if you're online, you can put your questions into chat, and Katerina will answer them. If we run out of time, we will get back to you here in McKinney. If you want to come up and ask questions of Katerina through the microphone. But first, let me just mention two other things. One, there's a question here about Native Plant Week. I did look it up. It is the 20th through 26th. That was on the site. So I went to the NIPSOT site, and it said 20th through 26th. Where was it different? Because it's a third full week of October. So yeah, that's what NIPSOT had. And also, I think Katerina had mentioned it. She will be providing the PDF of the presentation, and so we will be posting that through Facebook. So, all right. So, questions coming from the virtual audience. Well, here we have Melanie's got the first question.
All right. I just had a question on parasitoids, like the gall wasp. Is that considered a parasitoid, even though it's not laying in another insect or animal, or does it?
It would be a parasitoid for the plant.
Okay.
But again, because in many cases, if it doesn't hurt in it's co-evolved, kind of like the monarch with the milkweed. Yes, it eats it down to the nub, but then the milkweed comes back. Same story with the parasite.
Okay.
Wasps, yeah.
So it can be a parasitoid on a plant too. That's what I
Yes.
thought. I just wasn't sure. Okay, thanks.
Yes.
Another question here from McKinney.
Hi, great presentation. This is Janice. I'm just wondering how, when you're doing your discerning of whether or not to release a biological control agent, how do you decide what it is that or test, I guess, what other species that it might impact in the environment that you're moving it into? How do you check that it's going to be okay in
Right.
its new environment?
Yeah. So usually it's probably going to be a collaborative effort with taxonomists that are familiar with that particular species or that particular family, and even maybe the class. And so they would have to do an extensive review of the related species that we have, let's say, here in North America. And probably they would try to bring those into the lab or into a greenhouse. I've not been part of a study like that, but so I'm just kind of thinking off the top of my head how it would be done. But you would want to probably try to test them on these species that are in the same groups, general groups.
Okay, and then Rodney had asked a question there in chat. I don't know if you see that, Katerina, about references that you recommend on this subject matter.
I can put together a slide with the references that I used to put this presentation together, if that would be helpful.
Please.
Yeah, and there is a book that, and I do believe I was able to download it for free, that's all about parasitoids. So if you are really interested in them, it will make a good Sunday read.
Okay, and then somebody asked about the recording. The recording will be available. The Zoom recording will be available in about a week after today. All right. Any other questions here in the room or in the virtual audience? Oh, we got another question here in the room.
This was most fascinating. I know so little about this topic, and it's a little overwhelming. So
yeah, it is, and a week, no, was it a year ago? It was a year ago. I was able to take a workshop, and it lasted a whole 2 weeks, and we did 8 to 4 every day. So if there are any of you that are really interested in getting more out of this, I can also refer to you to these workshops that are taught once a year in January for 2 weeks. So Rodney, I don't know if you would be one of those individuals.
I don't know if I have the time for that, but I would be interested. This is more like a graduate level course that you would need to take to learn, and that would only touch just a little bit of it, I'm sure.
Yeah.
It was fascinating, though. I did not know anything about this topic, and I stand corrected with parasitoids now instead of
well, and now you can look for them when you go to the cemeteries, or you go to you can actually, I mean, you can see many of them, especially the Braconidae, the Igmonidae. They're larger, but even the small ones, if you look closely, they are out and about, especially when in earlier in the day. So just keep your eyes open.
Now, are you doing any work with like the emerald green borer? Is anybody researching natural controls on those? I'm sure there are somewhere.
So are you talking about the squash borer?
It's one that affects the elm trees, I believe.
Oh, yeah. I
Emerald.
do not know if it's being worked on or not. That's a good question. I will look into it, and I'll put that as part of my answers when I email Tim. But that's a good question. I'm sure somebody is.
Emerald ash borer, that's it. Sorry.
Yes.
Wrong one. Emerald ash borer, not ash trees. Yeah, I know it's just been found in this area the last couple of years, probably 5 years. So
right,
spreading into this area. I was just wondering if anybody, A&M was doing research on that?
I don't believe A&N is, but like I said, I'll certainly look into that question.
I think Texas Forest Service may be doing some things with that, and it has been found in Frisco now.
Right.
Mm. So it's moved from Fort Worth to Frisco.
If you are using these biological controls like in a greenhouse, are the vegetables and are they considered organic then? Is that
Yes,
because you haven't used a chemical agent?
as long
by
as,
any
yeah,
means?
as long as chemicals or only organically approved chemicals have been used. But as you can probably guess, a lot of these biocontrol agents are very susceptible to to the harder pesticides. And so it's an education process to make, and also the greenhouse owner, or they have to trust the system. They have to and they need to learn again how to synchronize correctly so that they can release these agents in time. But more and more research is being done, especially for the multiple use of these biocontrol agents. So for example, in our case, we just finished a study where we were using Bavaria bassiana, which is a fungus. And we know it works well to control the whitefly, but now we're interested in releasing, for example, there's a small predator called a big-eyed bug. That's a very, it's a very good predator, but we want to know if the fungus would also impact the predator. And so those are the kinds of things that more and more information needs, or more research needs to take place before the greenhouse owners can fully move into using those strategies and not rely on chemicals.
All right. Any other questions, comments here online? All right. I don't know, Ronnie, you have anything? Thank you, Katerina. Thank you very much for
You're
your
welcome.
presentation.
I just want to
Thank you so
thank you
much,
so very
guys.
much for a very interesting topic and presentation. It's most fascinating. I need to learn more about this, and I want to thank everybody for coming in person and joining us online here. I think we had a total of like 40 people online and at least 10 in person, if not more. So thank you very much for an entertaining 1 hour of learning about parasitoids. So I appreciate everybody coming.
Thank you so much. Have a good evening.
Thank you.